So now it's time to talk about the interaction between selection and drift, which is very interesting and very important. Drift makes adaptation less predictable, but it doesn't stop it, is the take-home. The nicest illustration of this principle is again from your textbook. It's an account of another experiment with flower beetles, which we met earlier when we were considering lethal recessive mutations. There's a locus called B in flower beetles, where the wild type B+ allele makes normal black exoskeletons. This locus has a mutant allele, B, which makes brown exoskeletons in heterozygotes who are B+, or reddish exoskeletons, which can be distinguished from brown visually, which are the homozygotes, BB. The mutation, the B mutation, is also deleterious. Selection therefore favors the B+ allele, but its progress is less predictable in smaller populations. And this phenomenon, this behavior, perfectly illustrates the very general and important trade-off between drift and selection, or trade-off, I should say, battle between drift and selection. So these two panels from Heron and Freeman show the results of this lovely experiment. The y-axis in both cases is the frequency of the wild type B+ allele, the fully viable one. And what the experimenters did was start a number like 10 replicate populations with equal frequencies of the wild type allele and the mutant that makes these slightly lighter, redder, browner exoskeletons. So all the populations start at a B+ allele frequency of 0.5. They ran populations in parallel that were either very small, 10 adult beetles per generation in all, or not so small, 10 times larger, population size of 100 adults. The heavy black lines show the average allele frequency as a function of generations 0 through 20 in both cases. And the light colored trajectories are the allele frequencies of the individual replicates. In populations of the smallest size, n equals 10, here in the bottom panel, there were fixations of both alleles, both the wild type, better allele, and the mutant allele, B, fixed in less than 20 generations. So here's a fixation of B, the bad mutation, and here are some fixations of B+, the better allele. There were a few more of those. It looks like 3 of them, but nonetheless, 3 to 1, not a significant difference. And notice the trajectories are quite spread out, and they're all over the place. So it's not surprising that one of them fixed after about 7 generations. In the larger populations, 10 times larger, with 100 beetles per generation, the trajectories spread out a bit, but none of them has come even close to fixing after 20 generations. But in both populations, the average trajectory shown by the heavy black lines is amazingly parallel. They both increase at a rate that implies a selective coefficient against the mutant B allele on the order of 10%. So to understand this finding, which is exactly what we expect thanks to theory, we need to briefly meet the theory. This is explained in Heron and Freeman on pages 263 to 265. So your intuition by this point should be telling you that if the random allele frequency changes caused by drift are much more violent, vigorous, or larger on average per generation than the directional change that is caused by selection, then drift could overpower selection and allow deleterious mutations to fix in the population. That makes obvious intuitive sense, and we've seen it happen in a few of these experiments. So way back in 1962, the great population genetics theorist Moto Kimura derived this super important classic result, which is that for an allele with a selective advantage of size S in his notation, which I'll explain in a second, the probability of ultimately fixing in a population is this ratio, simple ratio of exponentials. Now you don't need to memorize this equation or anything like that, but for now just look at it and understand what it says. It gives an expression for a probability of fixing, which means the probability of eventually going to an allele frequency of 1, where the fitnesses of the three genotypes, big A, little a, and little a, whose frequency is Q, the fitnesses are 1 for the big A, 1 plus S for the heterozygote, and 1 plus 2S for the homozygote. Thus, in this notation of Kimura's, the little a allele is favored when S is positive, and conversely, the big A allele is favored when S is negative. What you should notice from the form of this ratio that gives the probability of fixation is that the population size, N, and the selection coefficient, S, always appear as their product, N times S, right? So it's upstairs is 1 minus E to the minus 4NS times Q, the allele frequency. The downstairs is the same thing without the Q. It's 1 minus E to the minus 4NS. So the population size and the selection coefficient are the variable parameter of this expression in addition to S, right? They together say what is the probability of fixation, which I've graphed here on the y-axis, as a function of Q, but they never appear separately. So that's telling us something really important that falls out of the equation, that the balance between selection and drift has to do with whether N is large relative to the reciprocal of S, the selection coefficient, because it's their product that matters, not either of them individually, given a certain present allele frequency. So let's start with the neutral case when S is 0. The equation in its limit implies that the probability of ultimately fixing for any allele is simply its present frequency Q, which makes obvious sense, and I've drawn that in as this red straight line, saying that when Q is near 0, the probability of fixing is near 0, and the same thing. When Q is a half, the probability of fixing is a half. When Q is 0.99, then the probability of going all the way and fixing is 0.99. So it's a completely fair lottery of fixation for a neutral allele. Now, when the product N times S is greater than 1 in absolute value, leaving aside whether it's negative or positive, selection tends to determine the outcome. That's what Kimura's result says. So let's look at these lines I've drawn. For N times S is plus 10, or N times S is minus 10. If N times S is a number like 10, then the little a allele is favored, and once it gets to any substantial allele frequency, let's say 10%, then its probability of fixing is something like 90% or greater, right? So once it's into the population, it's basically got it. It's going to go to fixation, almost certainly. And conversely, if the product n times s is negative, which is to say s is a negative number, then the little a allele is selected against, big A is favored, and so it doesn't stand a chance of fixing until, on this curve, it's already almost there, in which case maybe it stands some chance of drifting to fixation. But when, conversely, the product of n times s in absolute value is much less than 1, that would be these curves near the red line of neutrality. For n times s of the order of a tenth in absolute value, the alleles are going to act for all practical purposes as if they were neutral, regardless of how large the selection coefficient is. That is, regardless of how different the fitness of their carriers is, they will act as if neutral if the population size is small enough that the product of the fitness difference between the alleles and the population size is only of the order 1/10. Okay, and then n times s on the order of 1 in absolute value represents this sort of transition between neutral, effectively neutral behavior and behavior strongly determined by selection. That's the take-home. So small fitness differences will determine the evolutionary fates of alleles in larger populations, but larger fitness differences will be required to overcome drift in smaller populations. And as you can imagine, this principle is the subject of a lot of worrying by conservation biologists who are trying to secure the long-term evolutionary future of very rare species, which are rare because they've been pushed into a small remaining patches of the habitat that's suitable for them. Their populations being small are subject to having harmful alleles fix in them by drift overpowering selection. So that's a bit abstract. That you may be scratching your head and going, what happened? Well, luckily with this video, you can go back and look at it again after you've thought about it or read some of the material in the textbook or whatever. Maybe this will help also. These are movies that I made by simulating these scenarios on my computer. So each panel in these displays shows 100 allele frequency histories for a given n and s and initial frequency q0. These are similar in spirit to those pictures I showed previously from the textbook. But there are more of them, and the histories run longer. Each history runs for 1,000 generations along the x-axis. Time goes from left to right, or until fixation occurs. Within each row of 3 panels, 1, 2, 3, the population sizes go up by factors of 10. So the left-hand panel is for a little population with 25 adults. Then the middle one is for a larger population of 250, and on the right, the largest with 2,500 individuals. The first case is the neutral control, where S, the selection coefficient, that is the difference in fitness between the A1 homozygote and the A2 homozygote, is 0, and Q0, the starting allele frequency, is a half. Okay, so they're all started here at an allele frequency of a half, and the alleles have exactly equal fitness. So not surprisingly, in the small population case, they all fix within 100 to 200 generations, and the fitnesses are as close as you like to 50-50, A1 or A2. No surprise. Likewise, in the larger population, where they haven't all fixed at 1,000 generations, but 81 of them have, and again, they are exactly half and half fixed for A1, half for A2, and it's clear the rest are going to fix equally both ways. In the largest case, by amazing luck, a few came close to fixing during the 1,000 generations, but none of them actually did, and they end the experiment all spread out along the allele frequency axis, but again with equal numbers above and below frequencies of a half, so they are going to fix ultimately 50-50 given enough time, just as the smallest population did. Okay, now let's consider a case with selection. Against A2 in the upper row, here S is 0.004, which in my notation means selection against the A2 allele. And in the lower row here, this S, the selection differential, is minus 0.004, which means selection is against A1 and for A2. So now what happens? In the smallest populations, where N is 25, and the product, N times S, is in both cases, in absolute value, much less than 1. It's a tenth, like those inner curves in Kimura's graph. Once again, they fix 50-50 for A1 and A2. In both cases, whether selection is against A2 or for A2, it fixes half the time. In the middle-sized population, where the product of N times S is 1 in absolute value, this transition zone, some fix for the disfavored allele, many more fix for the favored allele, but it's not absolute. Starting from frequencies of 50%, you can bet that the chance is greater that the favored allele will fix, but you can't be certain in any individual case. Something like 10 to 15%, it would appear, of all the cases are going to fix for the disfavored allele, but only 15%, not 50%, as in the small population. In the largest populations, it's an absolute certainty that the favored allele will fix and the disfavored allele will not fix. In the case where A2 is disfavored, Q goes towards 0. As it happens, half of the populations have fixed by 1,000 generations. The other half are still segregating, but it's clear that they are on a downward slope, and none of them is going to come back and fix. And conversely, it's the mirror image for the case where A2 is favored. Half of them have fixed by 1,000 generations, and the other half are going to get there in due course. These are the cases where the product of N times S is in absolute value a number much bigger than 1, and just as Kimura's theory says, yep, in these cases we can predict with great confidence that the favored allele will fix once its allele frequency is reasonably high. So what about inbreeding? Here's a connection with that. It can be viewed as extremely small population size, entailing, of course, extremely strong genetic drift. A child of half-siblings, as diagrammed in this figure from, again, Heron and Freeman on page 280, it's going to be homozygous at 1/8 of its genetic loci for alleles inherited identical by descent from its double grandparent. Right? For half-siblings, they have one grandparent that is twice their grandparent, and then two other grandparents that are once their grandparent only. The inbreeding coefficient for such a mating is an eighth, which means an eighth of all alleles are identical by immediate descent from the double grandparent. If grandma is carrying one of those deleterious recessive alleles we talked about in the recent lectures, that could be quite harmful if homozygous, but are perfectly tolerable if heterozygous. There's a substantial chance, like an eighth, that the child of the half-sibling mating will end up being homozygous for it, will be affected, will have seriously reduced fitness. That's in a very small scale, family level setting, exactly the same thing that happens in a small population where deleterious alleles, including deleterious recessives, stand a chance of rising to such high frequencies that they will be inherited in the homozygous state by individuals who don't necessarily have such an immediate connection through a double ancestor. That process increases homozygosity, as we said, including homozygosity for rare deleterious recessives, and there's lots and lots of evidence showing that that is bad for fitness. Here's a study of these fabulous great tits related, these are European birds related to our chickadees quite closely. Paris major is the species on an island, I believe in Scandinavia, where the adult population averaged around 60 breeding adults on average, and researchers had been mistnetting and banding and getting to know these adults by name for a number of years, so they could tell when a pair, a nesting pair, were half siblings with, actually full siblings would create offspring with an inbreeding coefficient of a quarter. Here's the inbreeding coefficient of half. These would be the nestlings who were the offspring of half sibs, and then 16th for double first cousins, and so on. So these are inbreeding states related to the researchers, or inferred from the researchers' knowledge of the actual pedigree relationships of the parent birds. Well, by this measure of fitness, number of eggs the pair laid that failed to hatch, on average, it went up precipitously as the inbreeding coefficient increased, which is to say, as the effective population size of the ancestors of the eggs got smaller, and the force of drift became, for all practical purposes, that much stronger. And here's a graph we looked at recently in a slightly different context. Humans in about 40 societies where cousin mating is practiced, and where there's good data on rates of early childhood mortality, comparing mortality rate among children of unrelated parents to mortality rates of children of the offspring of first cousins. And as we said before, in the overwhelming majority of such societies, the evidence shows higher mortality among the children of first cousins. Okay, so here's our summary of this great long tour through migration and drift and its interaction with selection. Migration is the process that makes populations more similar to each other by mixing them. It's a strong evolutionary force, or a fast process, in the sense that it acts quickly. It increases genetic diversity locally by bringing new alleles into a population, which can happen in one generation even, and it can bring in ones that are not locally favored by selection. It may decrease genetic diversity globally by causing the fixation of alleles that are favored in certain subpopulations, but locally it tends to increase it. Drift is the process that makes populations and species more different by causing random fixation of neutral or effectively neutral alleles. It is strong and fast in small populations, where it causes rapid allele frequency changes, but weak and slow in large populations. So it all depends on the population size. On average, it does, over time, decrease genetic diversity as some alleles fix, others are lost from the population. And in small populations where drift is strong, fitter alleles will sometimes be among those lost, and deleterious alleles may be among those fixed. We humans had a long history of small effective population size back in our hunter-gatherer past, during which time we seem to have accumulated many deleterious alleles. These are, to this day, very important in medicine, and we'll talk more about this later. But now, today, in our modern, rich modern world, we have a humongous population size, so big that no deleterious allele can increase in frequency, or, well, it may be able to increase slightly in frequency, but it will never be able to fix, never ever, as long as we maintain a huge population. And most deleterious alleles are inevitably, if slowly, being eliminated. A huge, slow, but inevitable public health benefit that we get from being a very numerous species. The question is, can we keep it up? All right, that ends this long but action-packed tour through the second two of the evolutionary processes or forces, and a bit of integration of all of them. So look for fun on the quiz, which actually won't be hard, and we'll see you next time.